Nucleic acids. Nucleic acids were discovered in 1868 when 24 years old Swiss physician Friedrich Mischer he isolated a new compound from the nuclei of WBC. See, nucleic acids are also called nucleoprotein. Mainly, every eukaryotic cell containing true nucleus. In the true nucleus, nucleic acids, including proteins, are present in the nucleus. The component is also called nucleoprotein. The nucleoprotein contains protein part and nucleic acids. Nucleic acids are also called nucleotides. The nucleotides again divided into two subunits. One is phosphoric acid and nucleosides. Nucleosides again divided into two major parts. One is carbohydrates and nitrogenous bases. Carbohydrates are also divided into two types, deoxyribosugar and ribosugar. Nitrogenous bases again divided into two major parts. One is purine nitrogenous bases, pyrimidine nitrogenous bases. Purine nitrogenous bases are adenine, guanine. They are double ring structures. And pyrimidine nitrogenous bases are three. One is cytosine, thymine, uracil. They are single ringed structure. In DNA, uracil is absent. RNA, uracil is present. Only DNA containing thymine, cytosine, pyrimidines. RNA contains cytosine and uracil, pyrimidine, nitrogenous bases. Let's discuss about the deep explanation of nucleic acids or nucleotides. It is the complete picture of nucleotides. Mainly nucleotides are composition of phosphoric acid, sugar substance and nitrogenous bases. Phosphoric acid chemical formula is H3PO4. Every nucleic acid containing phosphoric acid groups as a phosphate ions. It act as the phosphoric acid act as a backbone of the nucleic acids. Backbone of the nucleic acid. Every phosphoric acid connected with sugar molecules with ester bonds. Each phosphoric acid connected with two sugar molecules with two ester bonds. So these ester bonds are also called phosphodiester bonds. Phosphodiester bonds are present between phosphoric acids and sugar molecules. Mainly the sugar molecules are two. One is deoxyribosugar and ribosugar. If we observe deoxyribosugar, ribosugar chemical formulas, here it is. Ribosugar chemical formula is C5H10O5. It is in pentanone ring-like structure. Mainly it containing 5 carbons, 10 hydrogen and 5 oxygen atoms on these sugar molecules. And deoxyribosugar chemical formula is C5H10O4. One oxygen atom is eliminated from the ribose. That oxygen atom is eliminated from the second carbon atom of sugar molecule. So this sugar molecule, this ribosugar molecule is also called deoxyribosugar. Particularly we mentioned second carbon loses their oxygen. So we called 2-deoxyribose nucleic acid. 2-deoxyribosugar present in the DNA. Ribosugar present in the RNA. Both are sugars in the nucleotides. In this diagram, DNA nucleotides are there. Here it is. Pentanone ring-like structure, sugar molecule belongs to deoxyribose because on the second carbon of sugar, oxygen atom is eliminated. So this sugar molecule is also called 2-deoxyribose sugar. It present in only DNA. The sugar molecules connected with nitrogenous bases. The nitrogenous bases are connected with sugar molecules with glycosidic bonds. Glycosidic bonds are mainly two types. One is C1N9 glycosidic bond. C1N9 glycosidic bond present between sugar molecules and nitrogenous bases like adenine. See, adenine is the double ring-like structure. It is purine. Every purine connected with sugar molecule with C1N9 glycosidic bond. C1N9 glycosidic bond means on the first carbon of sugar connected with ninth nitrogen of purines. Ninth nitrogen of purines. This bond is also called C1N9 glycosidic bond. Sometimes the sugar molecules connected with pyrimidines with C1N1 glycosidic bond. C1N1 glycosidic bond mean first carbon of sugar connected with first nitrogen of pyrimidines. It is a second chemical bond present in the nucleotides. First chemical bond is phosphodiester bond. It present between phosphoric acids and sugar molecules. Second chemical bond present between sugar molecule and nitrogenous bases. They are again divided into two types. C1N9 glycosidic bond present between sugar molecules and nitrogenous bases, and C1N1 glycosidic bond also present between sugar molecules and nitrogenous bases. If C1N9 means they are belongs to purines. If C1N1 belongs to sugar molecules and pyrimidines. If we observed pyrimidines, purines are double ring-like structures. They are also called nitrogenous bases. Double ring-like structures are purines, adenine. Here it is in this diagram, adenine. Adenine containing one benzene ring, another one is pentanone ring. In this total purines, first carbon is eliminated and it is replaced with nitrogen. Third carbon is eliminated. Seventh carbon is eliminated. Ninth carbon is eliminated. And these all carbons are eliminated and they are all replaced with nitrogen atoms. And ninth nitrogen connected with sugar molecules. Between two nitrogenous bases, hydrogen bonds are present. Hydrogen bonds are mainly two types. One is double bond and triple bonds. Always adenine connected with thymine with two hydrogen bonds. And guanine always connected with three hydrogen bonds. Guanine connected with cytosine with three hydrogen bonds. Total in every nucleic acid, nucleotide, mainly three types of chemical bonds are present. One is phosphodiester bonds. Second one is glycosidic bond. Third one is hydrogen bonds. These are the chemical bonds present in the nucleotide.